All right, so now let's get to our list and discussion of organelles. Now remember, organelles are at that level of organization just below a cell. It's still in the chemical level, which is kind of crazy because we associate these so much with cells. But our organelles, by and large, are going to be membrane-bound subcompartments within the cell. We've got a couple of these honorary organelles we're going to talk about that aren't membrane-bound, but the cell's not going to be much good without them, so we include them in here. Now, many of these that we're going to talk about, the nucleus, the endoplasmic reticulum, the mitochondria, these all are membrane-bound structures. So as the plasma membrane distinguishes one cell from another, or distinguishes the inside from the outside of a cell, so will these membranes distinguish cytoplasm from our nuclear plasm, nucleoplasm as they call it, or the inside or the outside of the endoplasmic reticulum as we go through these. Now we're going to take each one of these individually, and then we're going to add a few more to our discussion as we move through our discussion on organelles. Now, I want to give you an appreciation of this as well. In your body, you remember we had cells, and then you put different cells together, and what do we get in our level of organization? Cells and what? Tissues. Then we get multiple tissues, and we make organs, and then what's multiple organs? Organ systems. So in the case of a cell, we're going to have several organelles that function in series. And so in that sense, we're going to organize several of these organelles into an organelle system that we refer to as the endomembrane system. Endomembrane meaning inside membranes. Now some of these are directly connected to each other, and some of these are linked basically in series. Much like when you look at the production, let's say, of your cell phone. You have companies that are going out and they are mining these rare earth elements out of the ground. They process them, send them off to another company. This other company puts them together into the components for the phone. They sell those components to the place that assembles the phone. So you've got a number of different factories that are doing different parts. Well, in the endomembrane system, we have several of these different stops along the way for processing material. But we're going to start with the nucleus. Because the nucleus, and we're going to learn more about it, it is a double membrane system. But the reason we include the nucleus here is because it is directly connected to the endoplasmic reticulum. The outer membrane of the nucleus essentially extends out and folds into what is the endoplasmic reticulum. So we link those two together. Now, the nucleus, we know, this is where our DNA is going to be contained. All of our DNA, all of our genes collectively together, we refer to as the genome for the human species. We're also going to have our DNA copied, and we're going to begin the production of our ribosomes, and we're going to get to those maybe not today, but on Wednesday. But that production all begins in the nucleus. The endoplasmic reticulum. The ER is where we're going to begin the production of proteins, and we're going to start synthesizing lipids that are going to be the lipid portion of many of our membranes. But again, included here because we have the direct connection. Now, as we begin the processing of proteins in the ER, they're going to be packaged into these little membrane-bound bubbles called vesicles. And often they're just referred to as transport vesicles. So the proteins that began to be processed in the ER, they're going to get shipped to the Golgi complex for further processing, sorting, and then shipping. And from the Golgi, again, we have transport vesicles that are going to move that material to its destination, much like FedEx. And in our illustration, our nuclear envelope is in purple. We can see the folds of blue. That's going to be our endoplasmic reticulum. Here in purple again, darker purple, that's our Golgi apparatus. And they're showing these little bubbles, in which case this they're saying is a lysosome, a peroxisome. Here's another little vesicle. But our transport vesicles are going to look much the same, just little membrane-bound bubbles.
Are you going to have those pictures of the test?
Yes, now some of these I have changed at the last minute to include them, and I'll make sure to upload. I thought, did I upload the chapter 4 part 1? It says new in the D2L. I'll make sure to get that up there. All right, because I added and changed some pictures. That was at the end of last week. Now, in addition to the simple transport vesicles, we're also going to produce, through the endoplasmic reticulum, further processed in the Golgi, we're going to have some vesicles that are called lysosomes. These lysosomes are going to be filled with enzymes. And these enzymes are going to be able to break down material. So as cells, much like our white blood cells and macrophages, as they engulf material, cellular debris, pathogens. And do you remember what we call that process of a cell bringing in material? What kind of cytosis is that? Endocytosis. When we make this membrane-bound compartment where we have trapped some food, these lysosomes are going to fuse with that compartment and release those enzymes into that area so it can digest and destroy that material. So think of these almost like your digestive enzymes in your stomach. Now we're also going to have some that are called peroxisomes. And these peroxisomes are going to be filled with, among other things, peroxide ions. And these again are going to help to detoxify certain things in the body. But we're also going to see that some of our white blood cells make what are called superoxide ions, and it's almost like hand grenades that they launch outside the cell to destroy pathogens or to destroy infected cells. So all of these, the lysosomes, peroxisomes, further transport vesicles, those are going to be important and sort of the end point for our endomembrane system. Remember, nucleus, ER, Golgi, and then these series of our vesicles. All right, so let's talk about each one of these a little bit more detail. We're going to start at the beginning with the nucleus. What's inside the nucleus? DNA. That's our genome. Surrounding our genome is this thing that looks like a wiffle ball. Everybody know what a wiffle ball looks like? It's this plastic ball that's got holes in it. A lot of times you use it to practice. You play with it as a kid. But when you look at our nuclear membrane, I want you to understand it's like a wiffle ball within a wiffle ball because it has a double layer with a space between. And it's often thought that somewhere in the evolutionary past, there was a cell that wasn't really good at maintaining its genetic material, and another cell that was really good at maintaining its genetic material but didn't quite, able to do anything else. And so they decided to set up a partnership. And as this cell that was really good at maintaining its DNA, it just simply came into the other cell so that they could set up a symbiotic relationship. Now, as this bigger cell engulfed the smaller one, it wrapped its own membrane around the original small cell. That's how the theory is, or hypothesis, is how the nucleus ended up with a double membrane. One was the original membrane and one was brought in by the host. We're going to see that with another organelle. And in plants, chloroplasts are often thought to have evolved the same way, also have a double membrane. But again, with the wiffle ball, we do have pores. These are highly complex structures. These pores, there are over 100 proteins in each pore complex that regulate the trafficking of material in and out. In fact, material in and out of a cell is much like you calling an Uber driver. There are proteins called, this is freebie, you don't have to write this down. There are proteins called important and exportant that must bind with a protein that's trying to get into the nucleus so that it can get past the transport proteins that guard the pores. The same way, if something's trying to get out, it has to bind to exportant, and exportant's like the key that allows it to get out. So these, just understand, these aren't just structural pores. They're highly regulating what goes in and out of the nucleus. Now, this is a very high electron microscope picture of a nucleus. You can see this line, and if you could look closely, you would see there were two dark lines. That's the double membrane of the nucleus. We have our genetic material inside. And then right in the center, often it's in the center of the nucleus, you see this really dark spot. And this really dark spot is called the nucleolus. And in the nucleolus, this is where you're beginning to produce and assemble the components of the ribosome. Now, ribosome is going to be part protein, but mostly the ribosome is going to be RNA. And the production of RNA begins inside the nucleus. So as we go to make our protein, which we're going to talk about maybe Friday or next week, we have to have 3 different types of RNA, and all 3 of those types of RNA begin to be produced in the nucleus. And so you see this dark spot because there's so much stuff that is being produced. It just, light can't pass through that as easily, so it looks like a dark spot. Now, we're going to go ahead and begin to use some terms that you have to get familiar with. Transcription and translation. Transcription is making a copy of RNA from DNA. Transcription is making RNA using DNA. Now, I like to think of transcription in this way. It helps me remember. Whenever you see a courtroom and you see this person on this thing just tapping away, what is that person's job? That's the court reporter. Is she just taking shorthand notes? She or he, sorry, didn't mean to be sexist. She or he, what is she typing? Every single word that is spoken. So she produces a what of the courtroom proceeding? A transcript. The language spoken in the courtroom is the same language that she's typing into the machine, using the same alphabet. So if you are getting a word-for-word copy using the same alphabet, that's a transcript. Now, we're going to put that together into one of our RNA molecules. It's going to be a copy of the code. And that copy is going to be shipped out to the cytoplasm, where we're going to use that information to assemble our amino acids in the correct order. So we take our RNA, which is going to be the message, and we're going to use that to make a protein. So in our RNA molecule, what's the alphabet for RNA? It's the same alphabet for DNA except for one letter. What's that alphabet? What are the monomers of our DNA and RNA? Right, those are the letters, but remember that's nucleotides. Nucleotides, that's the alphabet of our nucleic acid. What's the alphabet for proteins? What's the monomers of proteins? Amino acids. So do you see how we're changing the alphabet from nucleotides to amino acids? And whenever you change one language to another, what is that? It's a translation. You have to have a translator when you're going to a different country, or you use Google Translate. So when we get to the cytoplasm and we're using our RNA to assemble proteins, that's translation. So maybe thinking about the alphabet can help you keep straight. What is transcription happening in the nucleus? DNA to RNA, same alphabet, nucleotides. When you get to the cytoplasm, we're changing the language from nucleotides to amino acids and proteins. Does that make sense how they came up with those terms, transcript, translate? So in the nucleus, this is where transcription happens. Now, as I mentioned, and you can see the two layers. They've sort of outlined them in white. You see all the pore complexes on our nucleus. There's the nucleolus. On the inside membrane of our nucleus, we actually have proteins that can bind to DNA and help organize the DNA within the cell. Bless you. And then, as I mentioned before, this outer membrane, you see it sort of folding out in that illustration there and there. They've cut that off. But those are the extensions of the outer membrane of our nucleus that then folds and becomes the endoplasmic reticulum. And since that's the case, you can see that this space between our inner and outer mitochondrial membrane, that's going to be continuous with the lumen of our endoplasmic reticulum. So it's difficult to separate what's nucleus and what's ER until you look at the position within the cell. You can fairly easily recognize the nucleus, and then sometimes all these other folds that are our endoplasmic reticulum will look completely separate. But understand they're really not. So I love me some amoeba sisters. And so here we have the nucleus containing our DNA. And you notice all these little magical dots that the nucleus is spitting out. That's going to be the RNA. And so a lot of times these little simple cartoons, they do have quite a lot of information in them. And so that is our symbol of the nucleus and what the importance of the nucleus really is for the human body. All right. So now, in the endomembrane system, we go to the nucleus, to the endoplasmic reticulum that is directly connected. Here in this slide, we can see those bridge-like connections between the nuclear outer membrane and these folds of the mitochondria, of the folds of our endoplasmic reticulum. Now, in the ER, I'm going to say ER a lot because that's just easier to say than endoplasmic reticulum. In the ER, understand we have two different types. And they're illustrated on the screen. One has got all these little pimples all over it. All these little dots, these little bumps. That makes the surface look rough. And so we will have the rough endoplasmic reticulum versus a portion of the membrane that doesn't have the bumps, which looks smooth, and so we call it the smooth endoplasmic reticulum. So first, we're going to discuss the rough endoplasmic reticulum, or the RER. Lowercase r, capital E, capital R, that's usually how that's written. And so all of these bumps on the surface of the endoplasmic reticulum, those are going to be our ribosomes. And we're going to cover that next, so it's hard to discuss one without the other here. But the ribosomes are going to be the protein factories. Ribosomes are our protein factories. And so by having all of these ribosomes on the surface, this is going to be the beginning of protein production for proteins that are going to be secreted outside the cell, or proteins that are going to be somehow incorporated into the membrane. The plasma membrane, the nuclear membrane, the mitochondrial membrane. And we can also think of lysosomes peroxisomes. They're not going to be secreted outside of the cell, but they're going to be secreted into these vesicles that have our debris or pathogens. So in a loose sense, that's secretory material as well. Now, the other thing that happens as proteins begin their formation on the ribosome and they're extruded, much like, have you seen a pasta maker? It extrudes the pasta. Well, as the protein gets extruded into the lumen of our ER, it's inside the lumen, between the membranes, where proteins begin to fold and they begin to process. And one of the major things that happens in the beginning stages is glycosylation. Glyco means what? Sugar. So what do you think glycosylation of proteins means? The addition of sugars to our proteins. Now that begins here. That's not the principal function of the ER. Protein synthesis for secreted and membrane proteins. That's primarily what we're doing. But we start putting these foundational carbohydrates on in the ER. So glycosylation begins in our endoplasmic reticulum. So here we can see our rough endoplasmic reticulum. We've got all of our little factories, the ribosomes. You can see amino acids in, and there we're getting this chain of amino acids coming out, which is our protein. And here we can see a little, almost like a time-lapse depiction. We can see our ribosome beginning to extrude a protein that binds with the endoplasmic reticulum. The protein continues to get extruded inside and is folded until finally the protein is finished. The ribosome pops off, and there we have a protein in the center that is destined to either be secreted, become a lysosome, a peroxisome, or some other internal vesicular protein. Now, we'll get to much more detail on that. Right now, we're just getting the idea of what all of these organelles do. And again, what is the term for protein synthesis? Transcription. Using the RNA language and translating it into amino acids and assimilating them into proteins. Now, before we get to ribosomes, smooth ER. Smooth ER is not about synthesizing proteins, but it is about synthesizing lipids. Now, it does synthesize steroids and it's detoxing other compounds. But just imagine, as the ER is producing these proteins, from the endoplasmic reticulum, we've got to get that material to the Golgi. That's the next step. And so we're going to form these little vesicles off of the endoplasmic reticulum, these transport vesicles, that are going to move the material to the Golgi. So what's happening to the ER every time you bubble off a little piece of its membrane? It's going to get smaller and smaller and smaller because it's losing membrane, right? Because that transport only goes one direction. It goes to the ER. It doesn't come backward. So as you're transporting material, you're losing membrane and you're losing lipids, phospholipids. So it makes sense you have to be making more membrane the more you transport. So more membrane for the endoplasmic reticulum as a whole happens at the smooth ER. And that membrane is lost in these transport vesicles that are leaving, headed for the Golgi apparatus. So we already mentioned RER. So the rough part. What makes the ER rough? Are our ribosomes. Now, our ribosomes, understand, they can be free-floating in the cytoplasm. If there is a cytoplasmic protein that has to be made, that's not going to be secreted, it's not going to be membrane-bound, that just happens on a ribosome just floating in the cytoplasm. The ribosome's bound to the ER, that's going to produce a protein that's secreted or transmembrane. So the ribosome, its job is going to be translation. Change that to translation. And as we get to translation, understand we're going to have multiple components. This is simply one. And think of the ribosome composed of these two units, much like your hands. As you curve and cup your hands together, there's a space inside. So this is going to be the molecular warehouse within which amino acids are assembled into proteins. And that's why the amoeba sisters have given this illustration. Here, this is going to be our messenger RNA. That's the code. You can see the ribosome is the little factory, the warehouse. We don't see the amino acids coming in, but we can see the protein being extruded out.